Welcome to pitFALL. My name is Umar Ghafoor and we are going to start a bioinformatics course. Basically, I'm going to divide my course into three parts or three courses: a beginner's level course, intermediate course, and advanced level course. Let us start our beginner's course. Have you ever thought why the dry labs are important, or how the DNA genealogy tests are performed to check the ancestry, or how the DNA paternity tests are performed, or how the DNA sequences are retrieved or compared or aligned, or how the phylogenetic trees are made from the comparing of the DNA sequences, or how the orthologs are found? If you have ever thought about these questions, then you are at the right place. We are going to answer all these questions in our course. Let us start. We will start with the history of the bioinformatics, and the first name came to our mind is the great god Mendel. He basically studied the role of the gene in heredity by performing his pea plant experiment, and he came up with the idea of law of independent assortment and law of segregation. But his work was left ignored till his death, and the work was rediscovered after 16 years of his death by the Krawetz. Moving down the ladder, we have two other names. First one, Frederick Miser. He was the person who discovered the nucleic acid in 1868, and the other names are Watson and Crick, and they proposed the first three-dimensional structure of the DNA in 1953. It was the revolutionary turn for the molecular biology. And the next one is the Frederick Sanger. He basically sequenced the protein for the first time in 1970s, and he first determined the sequence of insulin. And now the computer became the essentials in the molecular biology due to the large amount of data we have. Here you can see the sequence of insulin. Do you think that you can memorize the sequence, or you can compare it, or you can find a mistake? No, it is very tough for a human being to compare these sequences. But after the discovery of the Sanger's method of genome sequencing, many other proteins were also sequenced, and now we have a large amount of data, and we need to have a system to store it, or to retrieve it, or to use it. And the solution was given by the Margaret Oakley Dayhope when she was working at the National Biomedical Research Foundation. She compiled the first protein sequence databases, which was named as the Protein Information Resource. She has also aligned the proteins into families and subfamilies. We will talk about the protein families later. The next name to our list while talking about the history of bioinformatics is the Wattu god. He was the person who first prepared the DNA sequence database when he was working at the Los Alamos National Laboratory in New Mexico. He has also worked at European Molecular Biology in Germany and translated the first DNA sequence, which was also included in the Protein Information Resources. He was the person who had conceived the GenBank prototype in 1979. Talking about the history of bioinformatics, how one can forget about the Alvin A. Cabot. He was the early contributor of the bioinformatics who came up with the idea of biological sequence analysis in 1970s. And he has also released the comprehensive volumes of antibody sequences with between 1980s and 1991. So, what is bioinformatics? The term was basically coined by the Pauline Hogweg and Ben Hasper in 1970s. And what is the bioinformatics? It is basically the development and implementation of the aligned algorithms for the mining of the data which we have retrieved or which we have gained from the DNA and protein sequences. Basically, it is an interdisciplinary science which uses the techniques of the computer, stat, math, and biology to give the solution toward our biological problems. Bioinformatics usually include the computer programming as a part of biological studies, as well as a specific analysis of pipelines that are repeatedly used in particular fields of genomics. So here comes a question in our mind that why one should study bioinformatics, or why bioinformatics is so important. Let us draw a computational or statistical model to understand this. So we all know that our individual chromosome consists of a DNA double helix and each applied genome composed of 23 DNA duplexes. So a human being has 46 DNA, or we can say that 23 pairs of DNA double helixes, and that these 23 helixes contain estimated from 25,000 up to 30,000 genes, and the nucleotide database consists of billions of gigabyte bases, and the size of human genome is up to 3 billion letters. You should also know that the genome term was coined in 1930s, and it refers genes present in an individual. So genomic size ranges from 2 million up to billions. So we have a large amount of data which needs to be stored, and here you can see the exponential growth in data from growth of PDB from 1985 to 2007. You can see the peak of the graph, and that there is an exponential growth in the data. In 1982 you can see here the growth of the gene bank, and by the 2000 you have a large increase or growth in data. So you have a large amount of data which needs to be stored. So here we need the computer sciences to collaborate with the molecular biology, and these two fields combines to form a new rapidly emerging field, the bioinformatics. Basically, bioinformatics is need to organize the bulk data so we can retrieve them and we can use this data. We use many different statistical models, computational techniques, and algorithms designed by the bioinformatics. So again, you should have a clear knowledge that what is bioinformatics. Basically, it is not a single field, it is an interdisciplinary sciences which requires the knowledge of mathematics, biochemistry, computer science, and information engineering. Here is a diagram which clears the idea that you need a lot of knowledge from different fields in order to work properly in bioinformatics. So what are the activities we perform in bioinformatics? The first one is the identification of the candidate gene. Then there is the identification of single nucleotide polymorphism, or we can find the genetic basis of a disease, or we can study the unique adaptations of an individual and the relation of these adaptations with the genetic makeup of an individual, or we can induce the desirable characteristics into an individual, or we can find the differences between different populations. Or in the less formal way, we can say that bioinformatics tries to understand the proteomics or genomics. So simply we can say that bioinformatics is used to analyze or interpret biological data. It is used for developing algorithms, writing softwares and tools, and data processing. And last, we have the statistical evaluation of data. So what are the fruits of bioinformatics, or what we have got from bioinformatics? We have got the precise medicines and preventive medicines for the treatment of many genetical diseases, and the treatment and the practices have improved. And then we have the genealogy test to check the ancestry of a individual, and we have also the DNA paternity test. And the bioinformatics has played important role in forensics in last few decades. So what is the scope of bioinformatics, or we are talking about the question that why one should study the bioinformatics. So basically bioinformatics is digitalization of the biological information. As we have studied earlier that biological knowledge is growing exponentially and experiments are performed on the daily basis. So data needs to be processed, data needs to be stored, data needs to be organized and analyzed, and there is a proper representation of the biological data, so one can have a good job opportunities in bioinformatics. And if we are talking about the Asian regions, there is a rapid advancement in bioinformatics in Asia, so one can have both educational or research opportunities in different fields, as the different institutes, both government and private institutes, are offering different opportunities. Here we have some bioinformatics databases which you can use. Let me explain some of them. Here is the GenBank. You can use GenBank or UniProt to retrieve the biological sequences or to analyze the biological sequences by using the different tools. And we will explain these tools later, like FASTA and BLAST and different types of BLAST. Here is the protein database bank, which is used for the structural analysis of the protein. Basically, it is a large database which contains the 3D shapes of the proteins and nucleic acid, which is used for the researchers. And here is Interpro or PFAM, You can use them to find the protein families or for the motif finding. And the next tool is GenoCAD. GenoCAD is a tool used to design the synthetic genetic structures. What are the genetic structures? We will talk about it later in our course. And what is the sequence read archive? Basically, it is used for the next generation sequencing. And the last one we have is Kyoto Encyclopedia of Genes and Genomes. It basically provides us the understanding of the biological system or the utilities of a biological system. Here are some biological bioinformatics tools. And there are two types of basically tools, open source softwares and web services. Here is a list of some open source softwares. We will talk about these softwares in the practical videos, and here are some useful websites you can use to retrieve the data, you can use to compare or analyze the biological data. This was all it. These are some reference books you can use. I will put the links in the description. These are some web references you can use. This was all it. Thank you so much. Please like, subscribe and share. Thank you so much.